Welcome to FlowStars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry, brought to you by Beckman Coulter at Bite Size Bio.
In the latest episode of FlowStars, I'm joined by Ryan Brinkman, Professor Emeritus at the British Columbia Cancer Agency. And in its wide-ranging conversations, we discuss the clinical impact of bioinformatics and how to get data without a wet lab.
If I'm going to do bioinformatics as my career, I'm going to want data well standardized. I want lots of data because I'm not going to have a wet lab. So I'm going to be reliant on others for data. I kind of got this idea back from my PhD that you can get a lot of data from other people. They're really, really cool thing.
The clinical impact of bioinformatic data.
But also I had twice as much data as you'd ever had before. And one of the things that was interesting was in this disease, we know that the size of the CAG repeat is associated with your age of onset. And so the protein expands, this is a CAG expansion. And if you have over 36 repeats, you're going to get Huntington's disease at some time of your life.
And what really motivates academics?
What we do it out of love for what we do. And that's a very different motivation. And I don't know that that's something you can teach. That's something you have to feel.
All in this episode of FlowStars. Hi, I'm Peter O'Toole and welcome to FlowStars. Today I'm joined by Ryan Brinkman from the British Columbia Cancer Agency, or was out there. Ryan, how are you today?
I'm doing fantastic. Thank you so much for having me here.
So, correct me if I'm wrong. You just said you're Professor Emeritus and you're retired.
Semi-retired. But by now, as of last night, my lab is the hugest it's ever been. I just hired 5 people into my academic lab since January, on top of the other 5 or 6 I had. It just doubled my size after I left.
Wow. Is that all on your salary that you used to have that is now gone to employ 5 new people?
Well, one of it is I left with a bunch of cash in the bank. And the 2, I have some really cool projects I'm trying to finish off. And 3, there is no 3.
Gosh, I'm going to ask a personal question now. You said no question was off target, so here's one.
Yeah.
What are you?
Today I'm 56. Yesterday I was 55. I just had my birthday.
Wow. Well, happy birthday.
Thank you. Belated. You're a bit late.
I was going to say I'm in the UK, but that's ahead of you anyway, not behind you. I could have got away with it technically. Retired at 55.
Retired, but then I started I just started my career. So January 1st, I joined Dotmatics, and it's the best job I've could ever imagine to be. Doing essentially the same job I had up until January 31st. So now I am a VP and research director of at Dotmatics, doing flow cytometry bioinformatics there.
Wow. No. So this is going to be interesting. We'll come back. Let's stay where we are, and we'll come back in history. So you're VP of Dotmatics. For those who don't know what Dotmatics is, very briefly, go on, give us a sentence on what Dotmatics does.
I should have this down. You've heard of? The story is it's a company that is helping very large organizations. Pharma are primary targets. Do science better. And so like everything today, data is a massive problem, and it's being generated more and more and more. Lots of variety of data, the volume of data. Software is a great solution for that. AI is a great solution for that. Dotmatics is a platform of companies that bring together scientific solutions that enable other research. So case in point, you may have heard of PRISM, previously known as GraphPad Prism. I use that in my PhD. That's part of the portfolio. Dotmatics is the electronic notebook, part of that portfolio. They have awesome 3 companies in flow cytometry, FCS Express, OMIC. My previous company, Cytometry, is part of that. That you no longer see that got subsumed into OMIC. They have a bunch of tools in the protein field, clinical trials, it's essentially, you can think of it as the Adobe of scientific software.
Okay, they changed their name what, 2 years ago-ish?
Yeah, so it was Insightful Science was the umbrella organization that started it, and then they acquired Dotmatics. And because of the name brand recognition on Dotmatics, that is a large part of it. Because nobody heard it, and nobody essentially knows what is it, Insightful Science, but a lot of people heard of Dotmatics. So that is sort of it's both the platform and the umbrella name for the whole organization.
So you retired from academia, but now
with a pension.
But taking up a new career, therefore.
Yes.
In the industrial side.
Yeah. And I wouldn't say it was a new career. It's a new way of doing what I've been doing my whole life, but at scale.
So let's take you back then. So obviously, what would be the best term? A flow cytometry bioinformatician?
That works.
Would be a
those are three very good words to describe my academic career anyway.
So where did all this start out? Where did you get into flow cytometry? Yes. Bioinformatics? Yes. It's not doing flow cytometry bioinformatics. Now that's getting very niche and specialized, and an area that is exploding at the moment. So we need more of it.
It's not something that I grew up wanting to be. So that all started when I was let go from my position immediately previous to this one. I had to find a new one, and had to stay in Vancouver because Vancouver, beautiful. And I had two places I interviewed at. One was BC Cancer, and the other one was a Genome Sciences Center. In Genome Sciences Center, I would have grown up to be somebody who looks at DNA sequence data, sequence bioinformatician. And I'm very familiar with that field. I worked at Washington University, working in the genome space. Like thousands of bioinformaticians were doing it at the time. And then I interviewed at BC Cancer, and they tour you around and introduce you to the other PIs there. And Clay Smith was an MD at the time, and he's touring me around the lab. And there's a bunch of computers out there, and they're all doing flow cytometry data analysis. Never heard of it. And he says, we do this gating thing. You might want to look into that. And I looked, because it's very hard and it's subjective and time consuming. This was back in 2002, I think. Yes, 2003. So that night I went home and I never heard of this technology. And I went to Google and I Googled flow cytometry bioinformatics. What is this thing that I could do? And I literally got about 4 hits at the time. And I went to PubMed, and I think there were something like 8 papers that had been published in 2002. And I looked at the papers, and they're not bioinformatics papers when you put the flow cytometry bioinformatics. And I thought, okay, I can go be another guy doing sequence analysis, but there's nobody doing this thing. I knew I loved bioinformatics. I knew I loved big data, computers, technology, algorithms. Not that I'm good at any of it, but just what I love being involved with. I thought, if I'm going to do something, I'm going to do something when nobody else is doing it, because I'm not that smart. And this is like it's a blue ocean kind of experience. And then I got super lucky. So then the first grant I ever wrote was a travel grant, which isn't really a grant. It was like write a paragraph. And it got me $5,000 to go to ISAC. It was in Montpellier. And everything good in my academic career is come as a result of ISAC and as a subset of that Cyto. Everything that's been fantastic in my life has been through ISAC. If you're in flow cytometry, if you're listening to this podcast and you're not a member of ISAC and haven't been to a Cyto meeting, you have missed out on a large part of your life or potential for greatness in your career. And so I went there, and I had the idea, if I'm going to do bioinformatics is my career, I'm going to want data well standardized. I want lots of data because I'm not going to have a wet lab. So I'm going to be reliant on others for data. I kind of got this idea back from my PhD that you can get a lot of data from other people and do really, really cool things. And so I went there with the idea, if I'm going to do this my whole life, I'm going to do something around data standards. So that's going to enable everything. And so I wrote to Bob Murphy and the head of the data standards group at the time. Sorry, I can't remember his name. Said, I don't know anything about this field. I'm a complete newbie, but I'm really interested and super keen. And would you guys meet with me? And these are like two, Bob Murphy was a luminary in the field. He's a head of ISAC. He, groundbreaking research. He wrote the first real paper on clustering of flow cytometry data, like when there was 3-color data. And in his paper, he says something really cool. Now that we have 5-color data, things are getting really complicated, and we're going to need new ways to analyze data. It's literally in his paper. It's like you look at that and you laugh now. But they sat down with me. This is the wonderful thing about Cytos. You can walk up to anybody. Everyone's so welcome. I guess I have very limited experience with scientific societies. ISAC is wonderful. And they sat, I have this beautiful memory of the two of them sitting with me. And they just wanted to hear what I can do. And they took the time when I knew nothing in the field. And they're like, yeah, this sounds good. We'll support you. And it kind of took off. And then I wrote my first grant was an NHR1. And I had a wonderful experience that I had 4 months of nothing. I was left to my own devices at the BC Cancer to find my own way, for better or worse. And so I had 4 months to do nothing but write my first grant, best grant I ever wrote. And so I wrote to the 4 people that I found on Google and saying, I want to do flow cytometry bioinformatics. And they all said yes, which was great. One of them was Adam Treaster, who was the head of Flojo. Perry Halland, who was at BD Biosciences. You may have heard of them. Michael Oakes. I found him. Sorry, Michael, I can't remember anything about what happened with your part of the grant. And then the guy who I wrote to, bad thing is that he left after he got the grant. And so I was like, oh my God, the guy who was gonna be my grant left. Best thing that happened, Robert Gentleman took his place. Robert Gentleman invented R and bioconductor. All of a sudden I have Robert Gentleman on my R1 to do flow cytometry for bioinformatics. The way things happened could not have been any better. So I had the man who invented R, a guy at BD Biosciences, and the CEO of Flojo on my first NHR1 grant, and I knew nothing about flow cytometry. So I was off to a good start. That's how I started my life in flow cytometry.
I love it. I also like the irony that you've got TriStar. So thanks to Flojo, and here you are now working for FCS Express, kind of their big rivals in
yeah. Well, I'm collaborating with people who are direct competitors, right? Even today, competitors on paper. So we're doing this project on. So we're doing a workshop on AI and flow cytometry and ML and flow cytometry for clinical diagnosis. I'm there. Sherry Green from OZ is there. Camila, who I love dearly, also a competitor of Dotmatics, is there. We all want the same thing. We all, and when I work with pharma, all the pharma want the same thing. So I have really close collaborations with BMS. They're really happy to work with other pharma to make the world a better place. And again, my whole stick is collaborative science, and we can compete, but that doesn't mean we can't collaborate.
Yeah, I am thinking, we run hands-on flow courses, and we have the different companies all come and support it together. And they are together, they talk together, lunch together, because they're teaching together. They're not teaching against each other, they are collaborating for the teaching side. So it's a big, and you mentioned teaching, and I only got a few photos, and you got a montage of collaborators. I don't know where to go. If you're watching, if you're listening to this, you'll have no idea what this picture is, but it is a picture of, 1, 2, 3, 4 different groups, all presumably of different collaborative projects.
It's all the same collaborative project. So this is all the FlowCap meetings that we have. So FlowCap was a series of competitions, for lack of a better word. Is more about understanding, a series of meetings to understand the state of the art of automated analysis of flow cytometry data. So Nima, who was the PhD in my group, who is now an eminent scientist at Stanford, was the first author on a series of those, on a couple of those publications, one of those publications for sure. where we're trying to understand the state of the art. And so we brought everybody together in the field, which is literally that is all of flow cytometry at the time. I think we did a really good job at reaching out and bringing people together as part of that work. Again, it was funded by BD. Jack Dunn, I love you. He bought pizza when there was 10 of us in the room of people who are doing flow cytometry and just trying to get the field started. Again, a lot of the early work was just trying to get the software in R developed. But it's all just everyone comes together just to bring the field forward.
Which is really important. Just going back, when you were 10 years old, what did you want to be?
Oh, older. I guess I didn't really have like I want to be an astronaut thing in my head. I didn't ever really know what I wanted to do. Even when I was in university, I think I changed my major 3 times during my
So what did you choose to start with then?
Biotech. I always loved computers. So when I was a kid, I had a VIC-20. That's when you had to put a little cassette. My brother worked at Digital. They no longer exist anymore. So when I went to summer holidays there, I could dial into their VAX-11. And that's when you dialed in. This is my start to computing. When you dialed in with the phone and you listened for that, like in Matrix, I was living that life. And then you hear, and you push it into these plungers to connect. And then so I could play Star Trek, like little green X's on the screen and move them around. Actually, before that, my first programming was on punch cards. I did programming in Fortran in high school. So I always loved programming. So I knew that was going to be, I never took any formal training other than in high school on that. But I always knew that's going to be part of my life. But I also loved science. I love discovery. The thing I like most about my job is being the first to know something. Like nobody else knows this. I have like my eureka moment in my PhD is like, this is super freaking cool. And I've solved a problem that people have been thinking about. It's the best. It's like a total head rush. The same kind of head rush you get when you're on stage. It's just shivers. So I loved science, but I didn't know what I wanted to do. So I did biotechnology. Long answer to a short question. It changed over a few times. And it wasn't until the 3rd year, I think, George Carmody, it was a genetics course. And then it was the magic dance of polymerase and DNA and how that all just works. Blew my mind. Like, how incredible is this dance of things that makes life exist? That it's so beautiful. And it's like this is science is a thing. I don't know what I was going to do in science, but I just knew that was my thing. Still like computing. And I guess my big break was after I did some database. After my undergrad, I did some work in doing some database stuff. But then they had a post for a job at WashU in Seattle, and a friend at the time ended up marrying. Fiona said, "Oh, here's this cool job in WashU. They're looking for bioinformaticians," and the bar was super low at this time to get into bioinformatics. Like, I knew how to run a SQL query like it was basically Access. It wasn't even SQL, but I loved computers, and that was enough to get me in. And then I learned how to do Perl programming and stuff. And then what happened is I was in an academic environment, and it was in St. Louis. So 2 things happened. What ended up through a series of things, was going to start a family. It wasn't going to be in St. Louis. Wanted to go back to Canada for various reasons, lifestyle being one of them. And I was surrounded by academics, and as somebody with just a undergraduate degree, all the cool people, all the cool projects were going to people with PhDs. Like, they're doing really amazing, cool. I want to do that. But at least in where I was, and for good reason, you kind of needed a card to say you're good enough to do some cool projects. And so I knew that I wasn't doing a PhD to get smarter. or to learn more. To me, it was just a calling card that would let me get to do cool shit. And that worked. Like I did my PhD on Huntington disease. Nobody have never touched Huntington disease again. It taught me, what it did teach me is how to do things. It didn't educate me in a domain that was ever useful. It taught me how to do science. It taught me how to do large collaborative projects. In my PhD, my whole reason I got my PhD is because my professor had two databases. One was a research database. It was on people who had Huntington disease. And he had his research database, there's one Access database, and he had a clinical database, which is, I forget what it was in. I think it was also in Access. And never the two were not connected. But his question is, can we mine this data to discover something new? He's like, here's some data, figure something out. And I thought, well, what if I put these two databases together? Let's try that. And so that was my PhD, put these two databases together and it wasn't that magical a thing to do. But also I had twice as much data as he'd ever had before. And one of the things that was interesting was in this disease, we know that the size of the CAG repeat is associated with your age of onset. And so the protein expands, this is a CAG expansion. And if you have over 36 repeats, you're going to get Huntington disease at some time of your life. These repeats can go up to 121. We can make this diagnosis at any time. So devastating disease. And so there's did a few papers on the clinical consequences of this diagnosis that people can have. It's really interesting stories. Like there's one person who had lived their life. Their parents have it. So you have a 50-50 chance of having Huntington disease. And they never want to get the genetic test. And so they live their life freely, without purpose, if you will, because they had this shadow over there, this looming horrible thing. And then they got the genetic test and they found they were going to be okay. Significant psychological trauma, which is the opposite of what you think you have. All of a sudden, you're going to be okay, everything's fine. And then they live their life without purpose. And then this devastating So anyway, so can we figure out this? So the thing was, can we figure out the age of onset of the disease? And so I had this large data set. I published a paper. We can get a pretty good idea when they have age of onset, but then I thought, okay, well, can we do better? And then there's a grad student, you can get away with a lot. So I wrote to everybody in the world who had a Huntington disease lab and said, can I please have your data? Everybody gave me their data. Part of that, and there was 40 labs around the world who had never heard of me. They heard of my supervisor, maybe that helped. But they said, here's all our clinical data. Now, it was easy for them to give me this data because it was super de-identified. The only thing I wanted was their size of CAG repeat and the age of onset. And now I had 10 times more data than anybody had ever thought of having in my life. And then we can come up with this essentially perfect way to give a prediction of the age of onset with like super tiny confidence intervals. Like by age 30, by age 40, you have a 98% plus or minus 2% having age of onset, which has profound implications for clinical work, because if you give a treatment to somebody, you want to know how long have you delayed their onset. But unless you have those unless you can make those predictions of when the onset could be, you could have never inferred when the onset would have happened. So you don't know how well your treatment is really working. So that was like, wow. So for me, that was the best thing. So I could see how my work would impact clinical science and development of drugs. And that has been my go-to thing since then, is using software to enable the development of drugs, of pharmaceuticals, of big data science. And that's what I'm going to do. That's been my whole shtick now in flow cytometry bioinformatics, is developing algorithms, big data, all of that. And with my own focus is enabling other people to use those tools to make discoveries at scale. And that is the amazing thing about being a bioinformatician for me, is I can hand off my algorithm to somebody else at some large pharma company who's like, we're working with, I guess they'll be talking about it at Cyto, we're working with Kite Pharma. And so they're doing this flow cytometry data analysis by hand. It's too time consuming, subjective. I'm just going on, stop me at any time here. they're doing this data analysis, trying to draw these circles around dots, which Clay Smith said, can you please solve this problem? Fast forward into my life to about 3 years ago, we have this problem solved. We hand it over to other people, and we're enabling to save patients that they weren't willing to give the drug to, because when they're doing their own CAR T therapies, because they're not certain about how they're doing the man analysis, it's subjective, and they're not willing to make that life death call that we're going to give this $300,000 drug to this individual because we're not sure how we're not confident of our gating, which is like, rah! But then we walk through with them with our algorithm, show how it's working. We parameterize it over a couple of months, and because we can explain our algorithm to them, because they get the ability to tweak how it works, they're super confident how it works. They look at 1,000 patients, they're super confident that it's giving the results. They're able to make their call on 3 people that they weren't able to make the call before. We got everything else right. Now they're rolling out in production across the company. And now we're saving lives at scale. And I have so many stories like this.
So,
that's why I am today.
So is that why you started your own company? To enable that?
Yes, that's exactly. That's a brilliant story. I did not start my company for that reason. So when I started the company, and that was one of the best things I ever did in my life, because I always had this feeling if I had been something else, to answer your question that you asked before, and I didn't know this later, I was like, I always wish I had done my own company. I didn't really care what that company was doing. I like coming up with ideas and having other people do the work. that's like the perfect job. I'm a lazy scientist. And so I thought if I was the CEO, I could come up with cool ideas to do stuff. Somebody else to carry it out. I can work on the ideas thing. And so when we invented this algorithm that does automated gating, flow density, this is in my academic lab, gave it away for free. And I knew it was perfect. It is perfect. It always works. There's nothing we've not been able to analyze awesomely. I'm not trying to sell you anything because we give it away for free. It needs parameterization. We can talk about some of the issues there. So you have to parameterize it for every panel. That's the downside. But when you do that, it's magic. And so I go give these academic talks. And one of the best things about being an academic in flow cytometry back in the day, there was not very many of us. I got invited to go everywhere because it's like something new that people want to hear about. So I've been all over the world to give these talks, and people come up to me after me and say, yeah, we'd love to use your algorithm, but we're not bioinformaticians. Can you help us? And it's like, yeah, but it takes, you got to grease the wheels, right? It takes money to do this parameterization. And I thought, and they always come to me after they've written the grant, after they collected the data. It's an afterthought. And so they don't have any more money in the grant. So it's kind of complicated. So I was doing that for a while, and then I gave a talk once, and some guy in industry came over to me. So they had the money. It was like a small little project, but it ended up being really difficult to get money in an academic environment because you need a contract that says we're going to deliver you this thing, and I'm not the one signing off on that. It's the institution, and so all of a sudden they're on the hook if Ryan Brinkman can't pull off this analysis. And so I did that once, and it ended up being like months of work to get that contract through, and through no fault of their own, but to go through them. And so I thought, okay, so is there a way I can get around that? I thought if I start a company, then I can work with academics. I work in industry kind of on the side, these little groups, and get the paperwork out of the way. I'll handle the paperwork and then subcontract all the work to my lab to do the work. I thought I'll help all these little academic groups and maybe some these small things that are coming up, I had this one thing, this small guy, there's like 2 people in the company. They mailed me their laptop to install the software on, and I mailed them the laptop back. It was to do some 5-color data, really simple stuff. So I made my little MySpace website with the blinking lights, so other people would find it. And I knew we could help pharma, but it's like I could help Genentech, I could help Pfizer and BMS, but I'm just a little professor guy, and I don't even know, like I can't call anybody to say we work with us, and it's like a few tens of thousands of dollars. It's not something that some person at the lab can just sign off on, and it's never going to happen. They're not my clients. It's just these little people I see that. but then I started getting emails from pharma, Google search worked, I guess. And it's like, oh my God, I'm working with pharma. They found my website, and it's not, and so then 3 years later, after 4 years, so 3 years of this company running, I kind of built up. By this time we had 6 of the top 10 pharma as our clients doing work, and I'd seen it's working. We helped Novartis getting Kymriah. It's like the first CAR T therapy. We got a really nice thank you for helping us with our FDA submission. And it's like, this is an incredible thing. Okay, this company thing is working. So maybe I should go back and learn out how to start a company. I just did it totally on the fly. It's like a one-man show. I was the CEO. I was the bookkeeper. I wrote and reviewed all the contracts. The only thing I didn't do was the accounting at the end of the year, because the tax man is the one who's going to bite your ass. And that's the one guy you got to make happy. But I did everything else. I thought, okay, I should figure out how to do this company thing. And I never thought the company would melt to something when I started. I made like 1 share in the company. When Justin left my academic lab to go to Paraguay, we just started a big project with Pfizer. I thought, okay, you can have this to get you. You can do this project when you go off to Paraguay to get you started, and then you can find your life there. So I only had 1 employee at the time, and I thought, okay, I'm gonna go learn how to do a company. I went to UBC. So if you're in a university, you want to start a company, go. They have project at every university do this. Stanford is great at this. UBC was really good. They have a little project called entrepreneurship at UBC. So I went to this course, did like the intro course. I thought, okay, now I get the basics, like know your customers. Like, oh, that would have been great if I could have figured that out and have your whole project plan and do all this. So now I go to like the intermediate course. And the day we're going to start the intermediate course, I get this email from Insightful Science saying, we're interested in your company. Do you want to buy it? I'm like, are you kidding? There is no IP in the company. There's 1 share, but yes. And so my first day at the entrepreneurship is can you tell me how to sell my company? It wasn't like how to get it. It wasn't the exit strategy. It's like I didn't know I needed one, but now I have one. And that started my links with Insightful Science. And that was about 3 years ago now. That was right at the beginning of COVID. The lawyer company, this whole thing is like, stuff can still work during COVID, because of the whole, I never actually got taken out to dinner. I think when you sell your company, they should take you out to dinner. They still owe me one.
Oh, you still haven't
got my.
Hey, come on. You're a VP. Take yourself out for dinner.
Yes.
Take yourself out for dinner and work and say,
yeah. But that was an incredible, just not, my whole life has been, what? That just happened? Without any planning.
So I'm going to flip, because you mentioned COVID and the impact of COVID, and I have just a few pictures. I have this one here. So this is obviously a cytometry part A paper, but this is about SARS-CoV-2. And this is with Andrea and Sarah, who are actually previous podcast guests. These are big cheeses. And Andrea was pretty fundamental in the COVID response and tracking within Italy on a national level. And of course, Italy for Europe was what
it was when the whole world went, oh my God. I mean, that's when I think the world realized, like, we're that word.
And it was right at the heart of the response, because that's for Europe, that's where it kicked off. Well,
it really was.
Certainly where it was found or most prevalent. Whether it was, let's let others argue about that. So this publication here. So tell me more about this publication.
This is a beautiful story. So this is right at the beginning of COVID. Right when lockdown started. Like, this is when you drive around, it was like the zombie movies. Like, the streets were deserted in Vancouver. You could drive down the street at any time of day and there was nobody out in the road. It was like one of the not the neutron bombs, because his car would still be left. It's like everyone was gone. And I got a email from a colleague who I never worked with, but again, somebody knows somebody who knows somebody, says, can you do anything with flow cytometry and COVID data? Like flow cytometry data and COVID, is there any link between those two? I'm like, probably. but this is just at the start of COVID. Is like, well, there's no data on it, but it's a disease of the immune system. Probably there's right. It's an infection, so there's probably somebody somewhere has probably thought of putting some data through a flow cytometer. So yes, and he's like, well, can you get some data? I'm like, well, I went to Google. Like, there's no, or PubMed or whatever. There's nothing that's been run through a machine yet, but I could get some soon. So okay, so can you come up with some idea that we can do something with flow cytometry? I forget what the pitch was something like, can you do something with flow cytometry data and put some data in front of people to help you analyze data? I'm like, no, because it's super complex, right? You have these panels, and you have to know what populations to define, and you have to draw these gates around them, and you have to train them on T cell and B cell biology, and you just can't throw that data in front of somebody and have them analyze it. That's stupid. But the pitch was, well, we can do this collaboration where we have this gaming company who is kind of get citizen scientists, gamers to analyze data for you. And I'm like, oh, a video game? So I'm a huge gamer. I'd heard of this. It's called EVE Online. It's a game I never played because I played World of Warcraft for years. The same premise. They hook you in with incremental rewards, and the only way you can how you win at EVE is to stop playing. And it's like but I know of it. It's a massively multiplayer online game.
I'm going to just switch because you also sent me. Oh
yes,
this one here.
So what we did is it worked. So now we took Andrea, we put him in the video game, and for the last 3 years, it's still going on today. We've had gamers around the world analyzing flow cytometry data for us 24 hours a day. I think we were at 360,000 gamers, give or take, hundreds of thousands of gamers, 360,000 accounts. Hundreds of thousands of gamers have analyzed millions of flow cytometry plots for me. I have the biggest flow cytometry lab in the world. So we solved that problem of having people who know nothing about flow cytometry data, analyze flow cytometry data. And I have a month to pull it off because I'm giving a talk at Cyto saying it worked. The jury is still out at this. So when they invite you to give a talk at a conference, I'm sorry, those people people at Cyto who future people who see my talk, when they invite you back in November saying, do you want to give a talk at Cyto, and you say yes, or when you write your paper or your abstract and you're not done yet, right? Often, and we're like nowhere.
How lucid your abstract? My abstract was, they asked me, I didn't know what I wanted to talk about. So actually we just published for a machine learning algorithm for imaging. I'm thinking, how am I going to shoehorn this into the Cyto talk that I'm giving?
Yeah.
Oh.
Yeah. So I knew I could talk about this, but you have to make it sound sexy. So like, oh, we've solved the automated gating problem. That's months from now. It's like, so now we're 4 weeks away. Stay tuned. It's going to work. Is it going to look super shiny like we solved the whole world in 4 weeks? No, but it's going to happen. And it's going to happen because we have so much freaking data. And it's good data. So it turns out you don't need to know anything about flow cytometry data in order to analyze it. And the only reason that worked is because of my work with flow density and the algorithm that we developed in the company. All you need to do to understand flow cytometry data is look at a single plot in isolation. And so my brain exploding moment is, so I had this other algorithm called flow type, and it's a way we do biomarker discovery, leveraging our flow density algorithm. So what we can do is we take every single population that you've gated and combine it with every other population to basically look at tens of thousands of populations. Then you separate your patients into treated versus untreated, sick versus healthy, and we'll find those that are different. And then I realized, and we're just going through all these bivariates in all combinations. And the algorithm doesn't know nothing about data or nothing about flow cytometry, he's just combining them. And a single bivariate plot, you don't need to understand anything about the biology to basically, if the goal is to just circle all the things of interest, you can circle all the bits. And once I had those two ideas in my head that came out of my algorithms, so that it's a Shakespeare problem. We have infinite amount of monkeys or gamers. We're just going to give them all the data and they're going to write Shakespeare for me, and the only reason why I did this, why I had to do this, is because, and I'm going to sit up for a second. As a society, as science, as flow cytometry scientists, as a society, we're horrible scientists. And this has been one of the things that I've been working on a lot. Because unlike most every other field out there where there's big science, there is no data sharing. There is a bit, but in flow repository and import, but not at the scale that you see for other technologies. Like every other, if you go to any other journal, if you're publishing a paper that has DNA sequence, you must submit your sequence to one of the repositories.
Yeah.
Every journal. The only journal in the world that says that is Cytometry A for flow cytometry. And so the only reason why I had to do this project is if I wanted to develop a machine learning algorithm to gate flow cytometry data, I could not do that because some people share their FCS files. Sure, they put them on flow repository. Nobody shares how they analyze that data. They might see one example, a couple examples in a PDF of a journal article. Here's how I drew my gates around one sample. That's not going to get anybody a machine learning algorithm. And so data sharing enables science.
So I've got to say with Eve, you're not the first to be using Eve. So on the Microscopists, the other podcast, Emma Lundberg has her own, and she has a super cool avatar. And Andrea has his super cool avatar. Do you not have an avatar on it?
They asked me. I'm not that, and I said no. Because
really?
Andrea, I know. I said no. Andrea's a rock star. Yeah, and so he's there because we got our first data set and so when this project started, it's like, it was understandably everybody wanted COVID data, and so he gave us the first samples that came out of Italy with COVID data, and I promised him we analyze it all for him. We had these dreams that we're going to analyze it in real time and give the data back to the scientists. It didn't work out, but we're going to have something better. We're going to have this machine learning algorithm. We're still going to do that, but in the meantime, we've been able to take all the data and we developed a machine learning algorithm that's going to work.
Really awesome for anyone listening, watching or listening. We've heard about the importance of data science around Parkinson's and how that's enabled Huntington's. And then we also had, you've talked about the analysis going into Kite and working with the pharma and Novartis and now COVID, the diversity of this type of research area. And it's not you've made it equitable, fortuitously, maybe made it equitable for fun, not because you wanted to make it. What gives you more? I think I know what your answer is going to be. What's the biggest motivation that makes you most proud? Is it the financial gain side of it?
Oh no! So I would have taken a job at mathematics for like, it's not a thing. It's being able to do things at scale, at enabling discoveries at scale is like I was in a relationship with a cardiologist once for a long time, and saving people's lives every day. Like people are going to die, goes in to save them. Great, fantastic. I loved what that meant, and meant a lot to them as well. But for me, the ability, like I can save like hundreds and thousands of patients, develop new drugs, enable new drugs that are save thousands of lives, all through algorithms and science is an incredible rush. It gives me purpose to get up. So it sounds corny.
No, not at all. I think this is really important because I think that there's always a fear that the best bioinformaticians will go into banking and the finance world because that's where big money is.
Yeah.
I think what's the best way to word this? Yes, some people will be financially motivated, and that's not wrong. And they'll enjoy the thrill of the ride of that type of industry.
Yeah.
But people in academia are not there for the money. They've got the same talents, the same skills, but are there to make a fundamental
100%.
difference. And you mentioned earlier the rush of seeing something for the first time, realizing something within the living world that no one else has seen and perceived before. It's like finding a new star in the galaxy. You have that on the bench. You have that on your desktop where you find that result that's new. That rush is huge. And to get data scientists, bioinformaticians, to see that it's not all about, don't just be attracted to the markets. Biosciences, your new medicines, it's where it's going to come from. Without the informatics, we're not going to get the best drugs.
The people who work in my academic lab and also at Domax couldn't agree more. They tell me that they fully well understood when they joined my academic lab that they could work figuring out insurance. It's a big data problem, or figure out how to get more people to buy my widget. That's a big problem. They are the people who I work with and at Dotmatics are motivated by our ability to enable science.
And do you know what? If only we could teach at the undergraduate level so well the relevance, the different impacts that people, and obviously we surround ourselves by people who are minded that way, but I think far more they don't even realize there's potential to do that.
No.
I think we wonder.
No, and it's not an easy thing to describe. It's easy to show on TikTok. I'm flash, look at my cars, money, happiness, association, right? It's a very easy thing to understand. But I think what we do it out of love for what we do. And that's a very different motivation. And I don't know that that's something you can teach. That's something you have to feel. I don't know that money works in that same way to motivate. It's different parts of brain.
That's good. I'm going to change tack. I'm going to ask you some quick fire questions.
Okay.
No, come on. I'm going to start with the flow cytometry quick fire. What's your favorite color?
Favorite color?
Yeah.
I like blue and orange. They complement. So actually, I'd say orange because my heritage is Dutch. So Oranje, the Dutch national color. But my don't get to wear a lot of orange. So if I'm wearing clothes, it's blue. If I'm flying the flag, it's orange.
I'm going to throw you a challenge to make sure that Cyto in your plenary you're wearing an orange top, and as a done deal for my plenary, I'm going to wear blue.
All right.
I think I'm going to get the better deal out of that. Okay. Are you an early bird or night owl?
Early bird or night owl. Is it a POW day? If it's a POW day, I'm up at 5 o'clock in the morning because my alarm is going off. Because I'm checking, my alarm is going off in the night. My brain alarm is like, check the snow state. So one of the best things about my job is when I start, best thing about my life actually was learning that I like snowboarding and mountain biking, coming to BC. And work-life balance is important. And so when I started at Dotmatics, it's a global company. And I talked to Brett and said, I want to be on New Zealand time. And I'm a better employee for working on New Zealand time. So that would mean I'm starting my workday late in the afternoon, right? But no, my workday starts at 8 o'clock because I'm first in line to get on literally the first in line at Whistler to go up and do some power runs. I do about 2 hours. Like I wake up at 5 because I'm so excited, get all my stuff on, have some breakfast, get out to mountain, hop on in the lift line. It takes about 10 minutes to get to the lift line. I'm snowboarding, uploading at 8:15, do a couple runs. I get to work and I am jazzed. I am so I'm pumped full of adrenaline. I'm excited. I'm not getting to work bleary-eyed. I've just done like 2 hours of powder or just hurtling down some double black diamond. And now I'm at work. Best thing about my job. But if I'm working, I'm working till I'll stay up till like 3 a.m. My best work is between like 10 o'clock and 3 o'clock at night. Because emails aren't coming in, I can focus. So I'm at both ends. I'm the spectrum.
Okay. So I've got to ask you really quick. When you get injured, how frustrated do you become?
It kills me. So I love mountain biking. So I have a plate here on my shoulder. Snowboarding, knees. I've wrecked my knees playing soccer. I've had ACL surgery on both. It's hard. Like there is a dependency, biochemical dependency that has built up in my head from adrenaline rush. And it's either from exercise or that rush you get after doing physical exercise.
Yep.
And when I'm injured, I am a shit to be around because I don't have that tick, 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 or like doing weights, like hitting the gym. And when I can't do any of those, it's horrible.
It is one of the downsides, I guess.
It is the downside.
PC or Mac?
False choice. It should be like a KDE or GNOME. What about Linux?
PC, Mac, or Linux?
Yeah. Unfortunately, Linux. Yeah, I would use Linux all the time, but then you have to tab out to do all the like Office kind of stuff. So it ends up being Mac. But if I could, I'd be running my own distro that I rolled. So
if it's Mac, McDonald's or Burger King?
Oh, then that's a false choice. That is for sure a false choice. So there's a on the way to Whistler. It's like a 2-hour drive. There's a McDonald's there, and it's like so we had to like you can't do that to yourself all the time. So it's pokey.
So you do McDonald's sometimes?
I do. It's a Big Mac, and the fries are so good, and a Coke Zero, which I don't know if you can get there. It's better than a Diet Coke. No.
No. It's the big. It's a breakfast. It's breakfast. If you get up early and you're traveling early doors, drop off for breakfast and get your caffeine kick there. Talking about that, coffee or tea? That
would have been a false choice up until I never drink coffee in my life. Can't get past the taste or smell of it. Never got a hang for tea, but then I discovered like a couple months ago, really just London Fog, which was invented in Vancouver. So you may not have heard of it. That's my new. It's a Earl Grey tea about halfway, and then lots of foamy milk on top.
Ooh,
it's because I'm a milk drinker.
Okay. Gosh.
So it's a good taste. London Fog.
The son drinks copious amounts of milk, and he writes. He operates. He's got his own operating system that he works in as well. He's also bioinformatics or informatics. Easier question, maybe. This is one that I need to know for Cyto. Beer or wine?
Wine.
Red or white?
Red. Bordeaux blends. Hervé, I'll never forget you taking me to some beautiful Bordeaux wines. Hervé Luce.
Oh. No, I'm a Californian Zinfandel person.
Oh, well, okay, but I am blessed to live in British Columbia because we have a little microclimate out in Kelowna, over the mountains. It's a desert, like cacti grow. We make some phenomenal reds in British Columbia. Like I would put them next to some California ones, like just top shelf. Very, very rich man owns like 5 or 6 wineries out there. I forget his name. He's trying to make the world's best Pinot Noir.
Yeah, we're going to have to move on because I'm getting thirsty just thinking about it. It's not a good place for my mind to go. It's not that type of day to drink. Chocolate or cheese?
Chocolate.
What's your favorite food?
Mexican.
And
because Mexican's always the same ingredients. You're just mixing up. It's like you got some guacamole, you got some sour cream. You just mix them up and see they're in a shell. It's on a plate. It's got some cheese. It's on nachos.
So who cooks at home?
I do. I do mostly, or I don't know. I like eating out a lot. So does that count?
If you cook at home, what's your signature dish?
Nachos.
TV or book?
Books, comic books are my go-to reading material. If I have the, but not the latex superhero. We're gonna, it's stuff like Right. There's the story. There's graphic novels, if you will. But they come in comic book form. These are the ones that get made into fantastic TV shows later, and people realize, oh, it came from a comic book. Become really compelling story. Saga. If you have to read one comic book in your life, read Saga.
Right. I've not heard of it. So now I'm going to have to look that up.
Look that one up.
Star Trek or Star Wars?
Star Wars changed my life. I mean, that's when I was that age where it's like, this is magic. I'd seen Star Trek before on TV, but then you see Star Wars on a big screen and it's like, oh my gosh. I had given the choice to go read the book or see the movie. And I thought, oh, if I read the book, I can keep rereading it again and again. Because I heard it. It's like, it was all in the news. Like, this is a great thing. Oh, I'll just read the book so I can get that story like 100 times. I can only go see the movie once. We didn't have a lot of money in West as a kid. That was like the worst choice I ever had. But then the library club went. So I got a chance to go with the library club. And it's like, oh my God, what if the book doesn't always match. Sometimes people say, the book is always better. Yeah, Star Wars, not so much.
It is, yeah, proper theatre at the cinema for that. And you know, I think you're the first person who's given me a good reason to prefer Star Wars over Star Trek. So I'll give you that one. What sort of music do you like to listen to?
Nowadays, a lot of electronic dance music, like Elderbrook, Bob Moses, stuff that you can dance to. It's just like it's just continuation from like the Cure and the Smiths, electronic music back then. Big alternative fan, alt rock, but now it's more like how.
Yeah, I think we're going to have to actually head out on at Cyto. So you said you listened.
Love clubbing.
You said you listened to a DJ podcast. Did you know that John Tiggs is a DJ?
I did not.
Aha. So with any luck, if we can get John to DJ, we should be made, and Andrea should be at Cyto. So we get Andrea and Sarah and everyone else up, and we can have a proper Flow Stars.
That would be awesome.
It is. That would be really quite cool to have. John, we're nearly out of time, and I've got loads of questions. Okay, when you retire, if you could do any job for a day or a week, short burn, what job would you like to sample?
But that's hard when you come in cold, isn't it? Like I'm gonna do a mix at a big rave, like with 10,000 people. Whoop, whoop, whoop. I come in, it's like when it goes dead, it was like, oh, that would be fun. So like two things: being on a stage in front of lots of people, huge rush, music, beef and everything, and then you then you do the big drop, and it was like. Yep.
Okay, so we've got to ask John to put a big drop in if we can get to DJ at Cyto as well. Oh, come on. Let's see if we get you up on there DJing with him.
Yeah.
Go. If you like, if you show you some ropes, to get you up there.
Yeah. Let me do the drop.
Do you know what? I think we have to keep these answers really short because we are up to 1 hour already. Okay. Come on, what out of all your career, when's been the most fun time?
Today. That's so easy. Because everything's been building up to now. I am living my best life. I could not be any happier, doing cooler stuff, having a better life than I am today. And the impacts that I'm making today are going to make. The people I work with are fantastic. The organization I work with is fantastic. The ability, the changes that we're going to enable across science are going to be amazing. Living my best life, doing my work-life balance is now there finally. When I started my company, it was like 24-hour day job. I loved it, but it wasn't right. I love being in that moment, but it wasn't good for me, and it wasn't good for my family and my kids. Now I'm in balance, and I have it's.
How do your kids reflect back on? How old are your kids now?
My son is 21 and my daughter is 16. He's in university now, second year.
How do they reflect back on
that was a hard time. That was a hard time. Something has to drop, right? I think this is a problem that we have a lot in our field. there's a lot of pressures when you're starting academia. I never wanted to be a professor. When I interviewed at BC Cancer, I forthright told them I do not want to be a professor, because I was fully aware through my relationship at the time with Fiona, that she was just starting out as academic. It is a 24-hour-a-day job if you allow it to be when you start out, because you're just starting out teaching, you're writing grants. It's just that you have kids in your lab that you're bringing up. It will consume all your time if you allow it. And then I started a company, and they were both going at the same time. It's not good. Something has to happen. And then they come here and the place is like a bomb went off, and then the kitchen's a mess, and it wasn't good for them.
But you must have justified that to yourself at the time. This is a good thing, the right thing to do.
So I just have to get through this. I just have to get through this. I just have to get through this. The light's gonna get better. Sometimes it takes a long, and it takes a lot. I think when all the professors have been through that, and it is tough, and you just have to get through that. But you have to. There's only so many times in the day, and that work-life balance is a real struggle in a field. And a huge shout out to all the moms who are professors who've done this. They're the primary caregivers, and that is a whole other burden. And so, just a huge shout out and respect to all the mother professors, CEOs out there. I cannot even imagine.
And on that, we are over the hour mark. I cannot believe that has gone so quick. But I will say, everyone who's watched and listened today at Flow Stars, just go and tune into Andrea and Sarah, for example, to start with. And John Teague's the DJ, the one and only. Super cool. Hopefully we'll see you at ISAC and Cyto. I will drop another one through the other podcast, Microscopies, because Emma Lundberg with their Eve project, and also Chris Lintott, who does the Zooniverse, another citizen science. So actually, if you want to make a difference and just enter into citizen science, you've got Eve, you've got Zooniverse. Lucy Collinson's podcast talks about drawing round electron micrographs, electron microscope images. Imagine how boring it sounds, but super cool. A difference that you can make, which is great. But Ryan, your energy, I think I have energy. Oh my goodness, I've been out energized on one of my own podcasts. That's amazing. Ryan, you are an inspiration to us all. Thank you so much. And I can't wait to buy you a glass of red wine at Cyto.
And the same in return. And that's how Cyto parties start.
Thanks very much, everyone.